everyone. I'm Arshiya Pathan. I refer review article DNA based vaccine. Its author name Kishwar Hayat Khan. Today we're going to discuss about what are a DNA based vaccine, their need, their types, and its working, applications, advantages, and disadvantages. Let's get started. What are a DNA based vaccine? Before DNA vaccine, we should know about a vaccine. Vaccine is a formulation, or generally we say is it is a preparation that is used to stimulate the body immune response against the disease. Let's come to the main point. DNA based vaccine are type of vaccine that use a genetic material from a pathogen, typically a virus, to stimulate an immune response in the body. DNA based vaccine use a small piece of DNA that encodes the genetic information for a specific antigen, which is a molecule that the immune system recognizes as a foreign. The use of DNA based vaccine has a several advantages over a traditional vaccines. DNA based vaccine can be produced more quickly at a lower cost than traditional vaccine, as the DNA encoding the antigen can be synthesized use automated techniques. Next, we see a need for a DNA based vaccine. DNA based vaccine offers several potential advantages over traditional vaccines. One key advantage is that they can be developed and produced more quickly and cost effectively. It can be designed to target specific variants of a pathogen, which could be useful in the context of rapidly evolving viruses such as influenza. DNA based vaccine can be effective against a wider range of pathogens, include those for which traditional vaccine approaches have been unsuccessful. The need for DNA vaccine arises from their potential advantages over the traditional vaccine. Next, we go through a types of DNA based vaccine. First, subunit vaccine. Subunit vaccine are a type of DNA based vaccine that use a specific part of pathogen rather than whole pathogen to trigger an immune responses. Subunit vaccines are designed to include only those part of the pathogen that are necessary to generate an immune responses, which can help to reduce the risk of side effects and increase the specificity of immune response. Take some example of subunit vaccine. Spike protein of severe acute respiratory syndromes, coronavirus 2, which is virus that cause COVID-19, and the malaria circumsporosite protein. These vaccines have shown a promise in preclinical and clinical studies. How subunit vaccine works? In the case of DNA based vaccines, the genetic sequence for the antigen is inserted into plasmid or viral vector, which is then used to deliver the DNA into the cell of body. Once inside the cell, the DNA is transcribed into RNA, which is then translated into the antigen protein. This protein is then presented on the surface of the cell, where it can be recognized by the immune system and trigger an immune response. Next, we go through a attenuated vaccine. They are a type of vaccine that involves the use of live weakened pathogen. Attenuated vaccine work by introducing a live weakened form of the pathogen into the body, which trigger an immune response that can protect against the future infections. Attenuated vaccines have been used for many years to protect against the infectious disease such as mumps, rubella, and yellow fever. However, they can sometimes cause adverse reaction in people with the weakened immune system or other underlying health condition. Let's see how it's work. When a person is vaccinated with a vaccine, the weakened pathogen is introduced into their body. The pathogen replicates within the body and trigger an immune response. The immune system respond to the pathogen by producing specific antibodies and T cell that can recognize and fight off the pathogen. Attenuated pathogen is still capable of replicating within the body. The immune response generated by the vaccine can provide long lasting immunity against the future infections. uh, there are several application: infectious disease vaccine, cancer vaccine, veterinary vaccines. Emerging infectious disease, personalized medicines. In limited clinical data, the DNA based vaccine are relatively new technology, and there is a limited clinical data available to support their long term safety and efficacy. Delivery challenge: DNA based vaccines require specialized delivery system to effectively deliver the genetic material into the cell. Immune response variability: DNA based vaccine can produce different level of immune response in different individuals. Stability and storage: DNA based vaccines are sensitive to degradation and require specialized storage conditions to maintain their effectiveness. Regulatory hurdles: the regulatory approval process for DNA based vaccine can be complex and lengthy. Cost: the development and production of DNA based vaccine can be expensive, which may limit their accessibility to certain population. Thank you everyone for your support and for listening my topic.